It's a very long title, so I
True that.
cut
Yeah.
it cut it a bit shorter.
Yeah.
Um, right. So what you can already do whilst the live stream is loading is share the link to the paper. And
Yes. Just a second.
everyone can log in and have a look.
Mhm. All right. Okay, so I've put a link in the meeting chat.
Fantastic. And then we're good to go. So welcome to Neuroccino on this Monday morning, the Oktoberfest edition. And today we have Robin present a paper on developmental dyslexia to us, and it's his first Neuroccino, so cheer him on and help him with all your questions. Robin, whenever you're ready, take it away.
Okay, so thank you for the introduction, Stephanie, and good morning, everyone. Um, okay, let's just see if I can share my screen. There it is.
We got the
Uh,
youngest member at Neuroccino this morning.
it,
Hello.
I can't seem to be able to share my screen for some reason.
Um,
Um, oh, wait.
so,
Wait, I've found out how to do it, I think. Can everyone see my screen?
yeah, we see the brain page.
Okay, wonderful. Okay, so then let's dive into the article. So the reason why I chose this article is because it's actually a very nice illustration of how certain advances in neuroimaging can actually allow us to sort of revisit some certain older theories, for example, about developmental dyslexia and the possible neurofunctional and neurostructural alterations that might underpin it. That at the time of the conception of those theories, we weren't able to actually investigate in vivo because, the technologies to do so simply didn't exist yet. So, what they basically did in this paper was to look at the magnocellular lateral geniculate nucleus, which is a subdivision of the visual thalamus, in developmental dyslexia, um, which, as you know, it's a very tiny structure in the brain. And so you really need high field, high resolution MRI in order to make sure that you're actually able to visualize the structure. Um, so this is a 7 Tesla MRI study that they've reported on. Um, so just a second, I'm just going to open up my annotated version of the paper. It's going to make life a bit easier for me. Okay, so as you all know, developmental dyslexia is a neurodevelopmental disorder with the main characteristic being that there are developmental issues in literacy skills. So these people with dyslexia struggle with learning how to read. It's actually quite a common developmental disorder. It's present about 5 to 10% of children, and it's also associated with with long-term consequences in terms of educational performance. So, for example, there's a higher dropout in higher education in people with dyslexia compared to neurotypical controls. And because of that, it's very important to try to understand what um, the neural underpinnings of this developmental disorder might be. And back in the 90s, there was this one theory that tried to associate developmental dyslexia with impairments in very sort of early sensory processes that were associated with the lateral geniculate nucleus of the thalamus, both in the visual and in the auditory processing pathways. And specifically for the visual pathway, there were some post-mortem studies that found abnormalities in the magnocellular layers of the LGN. Now, the LGN consists of two main subparts. So you have the magnocellular layer and the parvocellular layer, and they are associated with different sort of specializations in terms of visual information processing. So the magnocellular layer is typically involved in more coarse spatial image analysis, and it's particularly specialized in detecting motion and fast visual changes, whereas the parvocellular layer is more associated with color and fine spatial detail perception. And as I said, some post-mortem studies in developmental dyslexia found these morphological differences in the magnocellular layer, but not in the parvocellular layer of the LGN. Now, unfortunately, these studies didn't have the biggest sample sizes. So they refer to one study here that had only 5 participants. And yeah, back in those days, it wasn't impossible to run actual in vivo studies to compare LGN morphology and function between developmental dyslexia participants and neurotypicals. And so that's why this study now tried to revisit this idea, leveraging these advances in high field MRI. And so they managed to scan quite a large sample of 54 participants with and without developmental dyslexia. And they performed several fMRI tasks that were designed to localize the LGN, to sort of capture the functional properties of the magnocellular and parvocellular pathways. And they added in a third fMRI scan that they used as sort of a sanity check to really make sure that they have identified the LGN properly. Okay, so here you can see the different fMRI the different fMRI paradigms that they used. So the first one is this typical LGN localizer. It's basically it's just a series of lateralized checkerboards that were used to elicit brain activity associated with visual processing. Then this next paradigm was actually, again, sort of a visual paradigm where the visually stimuli were designed to specifically elicit responses in the from the magnocellular or from the parvocellular layers. And then finally, this motion experiment was used as a sanity check later on to make sure that the LGN, to make sure that the magnocellular voxels that they identified, truly were mostly driven by the response patterns that you expect from these magnocellular layers. So the reasoning is that these should be significantly more responsive to movement, visual movement, compared to parvocellular layers. Okay. So let's maybe have a look at how they defined the LGN. So in order to do this, they combined both anatomical and functional strategies. So first, they used a probabilistic LGN atlas that they fitted to each participant's individual brain. And within these regions of interest, these anatomical regions of interest, they then used their LGN localizers to obtain participant-specific regions of interest. And it's within those LGN regions, that they then compared the activity associated with the magnocellular and the parvocellular layers between dyslexics and non-dyslexics. And here you can see the results of their analysis. They compared the left and the right LGN separately, because there is also prior research that has suggested that there are a lot of laterality abnormalities in people with dyslexia. And there is this also very famous line of research that has indicated that people with dyslexia might be more likely to display atypical forms of hemispheric language dominance as well. So, for example, where the right hemisphere is more dominant for language, or where there is an overall reduction of asymmetry for language compared to neurotypicals. And so in this paper, they also looked at these types of laterality effects. And what they basically found was that in neurotypical brains, there is no asymmetry in activity associated with the magnocellular task in the LGN. However, in the dyslectics, there is a very clear and strong asymmetry. And it seems that the activity in the left LGN is slightly increased compared to the controls, whereas the activity in the right LGN is slightly decreased compared to the controls. So that's one of their main findings. We see an asymmetry that's present in dyslectics, but that's absent in controls. Oh, I forgot to mention this. They also looked at the myelination of the magnocellular and parvocellular parts of the LGN. And again, they compared the left and the right side. And what they found here was that, again, what they found here was that there is actually a reduced rightward asymmetry in people with dyslexia compared to controls, but only in the magnocellular LGN. Okay. Then later on in the paper, they also correlated the left-right asymmetry found in the functional response pattern of the magnocellular LGN with performance of the dyslectics on a rapid automatized naming task, or RAN task. So during rapid automatized naming, the participant has to say out loud as fast as possible a certain series of letters or numbers, or sometimes even has to name certain pictures as quickly as possible. And this type of task is something in which people with dyslexia often subperform. Even in pre-literate children, we can already see that rapid automatized naming can serve as a predictor of dyslexia or reading performance once they actually start to learn how to read. So the deficits in RAN are quite often associated with dyslexia. And so here they tried to correlate REM performance with these asymmetries in magnocellular LGN bold response. Now, in the overall group, they didn't find any significant effect. But then they split up the groups between male and female dyslectics. And when they do that, they do find a very strong effect in male dyslectics, an absent effect again in female dyslectics, although if you do look at the correlation value, it's the same trend as in the males. But I do think we have to be very careful when looking at these results because they're based on very small subgroups. So it's only 13 male dyslectics and 12 female dyslectics. So I do think we need to take these results with a grain of salt. Okay, now to summarize, this study actually was in a way able to sort of replicate the post-mortem findings of there being abnormalities in the magnocellular subdivision of the visual thalamus in developmental dyslexia. And it seems to suggest that these alterations are not only an important feature of this developmental dyslexia, but they might also be behaviorally relevant because they did find a correlation with REM performance, albeit only significantly so in the male dyslectics. Yeah, I think that's it.
Lovely. Thank you, Robin. Any questions from the team? In the meantime, I have a general question that may also go to other people in the room, but I was wondering while you presented, so they used a probabilistic atlas to map the LGN, which is a tiny structure, right? And it's extremely subcortical, as deep in the brain as you can get. And they obtained activations, fMRI activations from that region. I was wondering how good signal is that deep in the brain when it comes to fMRI activations. And that's a genuine question. I have no idea.
Yeah, that's a good question. I don't have experience with 7 Tesla fMRI data either, so.
Hervé, Mish, any one of you?
That was one of my questions. I was just looking at the methods. They have a resolution with the bold scans of 1.25 millimeters, roughly, and they applied a Gaussian filter of the same size of the voxels. So yeah, that's a good question actually. I mean, it's a really teeny tiny structure, so even with the raw resolution, that might be an issue. And yeah, 7 Tesla is a lot, but I'm not sure. It's
that's probably the reason why they've included all these sanity checks, because I did forget to mention, I think that they did find that the magnocellular layer that they identified, those voxels did respond significantly stronger during this motion fMRI experiment compared to the parvocellular voxels, which is in line with what you would expect the response profile of the magnocellular layer to be.
Something to be checked in more detail, I guess. I don't know who was first. Eva is on top here, so I'm going to go with Eva.
Thank you for the presentation. Maybe I missed it, but I was wondering how they measure myelination. Maybe this is common knowledge, but
yeah, I did indeed gloss over that. We'll have to look that up again, how they did that. Yeah.
I saw it. So they did it with a T1 mapping.
Okay.
Hang on, just a T1 to get myelin?
So you can do T1 mapping. It's not, yeah, that was one of my questions, that it's not really myelination, but you have microstructural measures when you do T1 mapping. I just saw the term T1 mapping. I don't know exactly how they did it, because normally it's a long scanning procedure, so I didn't check yet.
Yeah, it seems that they don't say it in the main body of the paper. It's somewhere in the supplementary material methods
No,
section.
if you look at the myelination, they not, yeah, they say it, but yeah, somewhere.
They say it somewhere. Yeah, maybe they say it.
No, it's some.
Here they say it.
Yeah, you see a quantitative T1.
Yeah.
Okay, interesting. I'll have a look.
Mish.
There is a large debate on how T1 mapping is related to myelination, so still.
Okay.
I've heard about the debate.
You contributed to the debate.
Yeah, so I was wondering, because dyslexia is a neurodevelopmental disorder. Conceptually, using an atlas have been defined in RC controls to map small structures like the magno and the pathophysiological portion of the lateral geniculate nucleus. So you're showing differences in bold and in myelin, but that might be a problem of alignment or division of those two territories that is different. Is that discussed in the paper, this alternate interpretation? Or are they under the belief that this is exactly the same brain that people will use differently, and that's why you're dyslexic? That makes sense. Morning for me as well.
Yeah, it makes sense.
So do they discuss it in light of? What was the question? Oh, you're looking for the answer. Yeah. Okay.
Yeah. Wait, I don't think I completely got the question.
Oh yeah, that's what I thought. It just came out like that. I'm sorry.
I thought it was more of a comment than a question.
No, it's more conceptual. It's a use and atlas that is defined in RC controls to quantify magno and pathophysiological portion of the lateral geniculate nucleus neurodevelopmental disorder. But and the interpretation is like, oh yes, there is a different in myelin and a different of ball, but that might be a complete different alignment because those brains, maturated differently on a different trajectory. And the division between the pavo and the magnocellular might be completely different for them. Do they discuss that, or?
No, they don't raise that point in the discussion.
Because the magnocellular portion could be like much bigger and overlap with what we consider standardly as pavocellular, and that will drive the entire results.
I'd be surprised to read that in the discussion, and also once you get to that conceptual consideration, would you still do a study using an atlas?
No, probably not.
Yeah, because it will probably also not differ between people with and without dyslexia, but also within groups. Like within groups of healthy individuals, you will have the same type of variability, right?
Right.
Yeah.
We gotta start from somewhere, I guess.
Yeah. And to be fair, they didn't just take an ROI, and they didn't just take an anatomical ROI and applied it just to everyone. They basically used an anatomical ROI to sort of constrain a more individualized selection of the region of interest that was based on subject-specific fMRI. So.
I think Hervé came before Eduardo.
No, No, I was the last. After Eduardo.
Okay, thank you, Herman. So yeah, thanks, Robin, for presenting. His reading is really interesting topic. My question is more general on the theoretical side, because so there are many theories of about the origin of dyslexia. So I was wondering if they considered other theories and how that would relate, because from what I remember, the magnocellular theory was sort of dismissed for a while as not so strongly supported by evidence. And so, yeah, just curious about that.
In this particular paper, they also only refer to the more general findings of atypical asymmetries in people with dyslexia. So, for example, here right in the introduction, they refer to alterations in a left lateralized language network. But even to my knowledge, even those alterations, they're still, up for debate. Like it's this thing. Like it's not always found by every study. But other than that, they don't seem to refer to other theories. No.
Okay. Yeah, that's interesting because there are at least a bunch of other theories. I'm sending in the chat a link to a couple of stuff if you guys are interested. There is one meta-analysis comparing different theories about dyslexia and also an experimental paper.
Okay.
Yeah, because the thing that made me think about it is that this kind of disorganization in the neural tissue are present also in other parts of the brain in dyslexic people. So yeah.
Yeah.
Moving on to Hervé.
Yes, thank you for the presentation, Robin. Just a naive questions about so they look at sex, so they look at men and female. So I was wondering if there is any hypothesis about that, or was it just an exploratory random results they got, or something to dig a little bit more?
Yes, so it's not something that came entirely out of the blue. In the sense that, for example, in the introduction, they say that, there are sex-specific differences in these alterations in the thalamus found in animal models. So they refer to animal models. But other than that, they, to my knowledge, don't refer to other reasons why they looked at this sex difference.
Is there any differences in frequencies, occurrence frequencies for between male and female in general, or not at all?
So I do know that dyslexia is a little bit more commonly reported in males compared to females. But that's the case for a lot of developmental disorders.
Exactly, yeah.
So then the question is always like, is this indeed like related to the actual underlying biology? Like, are there certain neurodevelopmental reasons or whatever why males may be more likely to develop dyslexia or other developmental disorders, or is it just something that gets noticed more easily in males compared to females? Or are there societal reasons for that rather than biological reasons? I don't know the literature well enough to know what the current consensus is about it. Maybe someone else knows.
Okay.
Yeah.
Thank you.
Yeah.
And final question to the youngest member in the audience.
So, yeah, thank you for this presentation. I just had a quick question. Like, they found this bold activation asymmetry in the LGN, but does this have any like cortical reflections, like any differences in the occipital regions? between
You mean,
controls. Yeah.
you mean like in terms of in terms of gray matter volume, things like that?
No, like bold activation differences
Yeah.
in the cortical regions, like in the occipital region. Do they find anything like that, or is it like not something that they looked into?
I'm not 100% sure that I fully understand your question.
Do you mean
what-
that they found changes in the subcortical structures? If these changes
Yes, the LGN.
are down the line at,
Mhm.
for example, the cortical level in the visual cortex?
Yes, I guess it's not something that they looked into.
I mean, they looked specifically only at the LGN, so they
Okay.
don't report anything else. Okay, yeah, okay, yeah. They don't report anything else about any other part of the brain. They only looked at the LGN here.
Okay, yeah, I was curious, like maybe other studies reported like activation differences in the cortical regions for between dyslexia.
Oh, yeah. So those studies exist. Like one of the most common findings is actually a hypoactivity of the visual word form area. So which is a part of the fusiform gyrus that is specifically more activated by written words and letters compared to any other visual category, stimulus category. And typically during reading and other visual tasks in people with dyslexia, we see a reduction in activity of the left visual word form area, which then results in a reduced hemispheric asymmetry. Yeah.
Okay, thank you.
Thank you. That's all the time we got. Thank you so much,
Okay.
Robin. I know it's
Well.
stressful to do the first neurotic. You did a very good job. So thanks for volunteering. And then a wee announcement before I let you go is that we were supposed to have a CNS talk this week by Andras Jakob from Zurich on fetal MRI. Really cool work. But he has to do a scan just at the time when our talk is, so we decided to postpone it until next week. So next week we're going to have Andras CNS talk on Wednesday 4 p.m. Do join us for that. And then all that's left to say is have an exciting week in science and see you on Monday. Bye.